Good morning, dear students. So we are here to study the chapter seed, and it is of chapter 6 of ICSC Concise Biology part 1 class 9. So we will be studying about seeds. What are they? So what do you understand by seed? Or first let us recap some basic point that suppose what are fruit. So as we all already have learned about this, so continuing with the chapter of seed structure and germination, we will come and understand in details about the chapter the seeds. Okay, so seeds, it is also we will be studying structure and germination. What I say, structure and germination of seeds. Before we talk about the germination of seeds, it would be appropriate to refresh your knowledge about the actual meaning of the term fruit, seed and grain. Okay, so as we will be proceeding towards the chapter, I would like to inform you all that I am following Concise Biology class 9 and it is chapter 6 over here. So I request you all to please open your book and keep it in beside this video. As I will continue teaching in the video, you go through and follow through the book, because that's how we can fight against the online situation or the online mode of studying. Okay, as you all are not in front of me, so I cannot have that eye contact, or I cannot make you explain in a offline manner. So we are going on in this particular format, in a video format, where I will try to teach you everything and make you understand everything, but you also need to cooperate a bit. So keep your book open and hopefully we will understand at the end of this video that what this chapter is all about. So moving forward, what is fruit? Fruit is the enlarged ripened ovary, the ovarian wall forming the fruit wall which enclose the seed. The fruit protects the seed and helps in seed dispersal. Example, mango, pea pod and everything. Okay, now seed is the ripened ovule. So what we understood? Fruit. What is fruit? So fruit is the ripened ovary. Now you all have already studied the previous chapter, that is pollination and fertilization and the flowers also, I hope. So you all know that which part becomes what. Okay, so fruit is the ripened ovary and seed is the ripened ovule. Okay, so seed is the ripened ovule. It contains an embryo which develops into a new plant. The seed coat protects the embryo from mechanical damage. Example, bean, seed, pea. Okay, now coming to grain. Grain is actually a fruit in which the fruit wall and the seed coat are fused. So fruit wall and seed coat, what are they? Now seed coat, suppose we have taken a mango. Okay, so inside the mango, what we find that there is a seed, particular big type of seed. Okay, now in any of the case, for example in maize and wheat, what happened? This seed coat, okay, this seed coat, these get fused with the fruit wall. Okay, and we cannot separate them. Thus we call that particular thing to be a grain. Am I clear? So moving forward, more about seed. It is a mature ovule after fertilization. Definitely after fertilization, because we all know that each and every of the fruit, it comes from where? From flower. And this fruit, when this get disintegrates, this forms a new plant, or this germinates. Okay, it contains a tiny living plant, the embryo. Okay, the embryo consists of radicle which becomes the future root. The embryo remains in an inactive or dormant state, but upon exposure to favorable condition, it germinates. The seed may also contain endosperm, which is a food material for the nourishment of the embryo during germination. Now what happens, my dear student? it contains of a tiny embryo. Okay, this embryo further on goes and it forms the new plant. Okay, the embryo remains in an inactive state. Okay, so see, we all know that germination or the seed, it can be preserved for a long time. Now the seed coat is so strong that it can actually fight against any of the in any of the adverse situation or adverse climatic condition. So the seed coat is prepared in such a material that the embryo inside the seed coat, it remains inactive and dormant for a long period of time. Moving forward, the seed can withstand unfavorable condition of temperature, drought, etc. Some seeds are known to remain dormant even upon 100 years or more. Okay. Classification and structure of seeds. Seeds may vary in size. Some are so small that they are barely visible to naked eye. Example, poppy seed, strawberry seeds, etc. So these seeds are very small and we cannot even look into it bare eyes. We need special aids, or we need at least a magnifying glass to see it properly. Okay, and otherwise we have mango seed, which is so small, so big that we can easily see look onto that, right? So some are quite large. Example, mango palm, as I already told. Largest seed are those of coconut and double coconut. So how many of you have seen double coconut? Okay, so it's a task for you all. Go and search double coconut in Google, or if available you can buy it from the market and have a look on that. Okay, so type of seeds based on the endosperm. So we are classifying here the type of seeds based on the endosperm. Okay, now types based on endosperm. So what are the type based on endosperm? Number one is albuminous, another one is exalbuminous. Okay, so types are what? Albuminous and exalbuminous. What are albuminous? Okay, in such seeds, cotyledon are thin and membranous and endosperm persist. Example, monocot albuminous seed, that is cereal, millet, palm, and dicot albuminous seed, example, poppy and custard apple. So what happens in such seed? Cotyledons are thin and membranous and endosperms persist. Okay, endosperms remains in that particular type of seed. Now what is exalbuminous? That is non-endospermic. We can call it as non-endospermic. Okay, and this one is endospermic. So what is exalbuminous? In such seed, the cotyledon store foods and become thick and fleshy. Monocot exalbuminous seeds, examples are orchids, and the things are given in the book. You can look from that. And what are dicot albuminous seeds? They are gram, pea, mango, and etc. So I hope this is clear up till now. So broadly, the seeds are of two kinds: monocotyledonous and dicotyledonous. Okay, so from here we can see that broadly its type, if we define them on their types, we will find that it is of two types. One is monocotyledonous. Monocotyledonous. Another one is dicotyledonous. what happens is monocotyledons and dicotyledons, we will learn about that. Okay, monocotyledon seeds contains one cotyledon, that is examples are what? Examples are maize, grass, etc. And what are dicotyledon seeds? That contains of two cotyledon, example pea, gram and bean. Okay, so from the name itself we can understand, here it is one cotyledon and here there is two. Okay, so the term mono and di. So moving forward, we have the bean seed. What is bean seed and what happens in bean seed, we will study about that. So student, we have came here with the diagram of the seeds. Okay, and these are all bean seed. These are all bean seeds. Okay, now this particular diagrams, everything what I am teaching over here, it is available in your book, that is Concise Biology, ICC Concise Biology Part 1 Class 9. You can follow that with page number 48, the bean seed. So before going on to the book part, what we will continue with is first the part of the seeds. Okay, now this entire seeds part, okay, this surface, this is known as cotyledon. What I have already told you about monocotyledons and dicotyledons. So this is cotyledon part. Okay, this is cotyledon part. Now this part, this particular part is known as hilum. Every of the bean seed have two parts. One is the convex side, another one is the concave. What is concave? When the thing is like this, or it is a cave like structure, so we call it as a concave, and when it is like this one, we call it as a convex. Okay, bulging out, we call it as a convex, and which is going deeper, that is known as concave. So now this one is the part of hilum, and this outer lining is known as testa. What? It is known as testa. Now if we just cut this part, okay, cut this part and see a other view of this particular bean seed, we will find that here is a small opening which is known as micropyle. Okay, which is known as micropyle, and this is testa, as I have already told. Now we are furthermore studying another segment or another view of this particular bean seed. What we find over here, see, tegmen is the inner side of this part, okay, and testa are the outer two part or outer two side. What is hypocotyl? Hypocotyl is this green part which further grows to or germinate to a new plant. Okay, these further germinate to a new plant. So this is called hypocotyl, and radicle is the tip. It is the tip which grows further to or which germinates and comes out of the seed and of the seed of the soil as a new plant. Okay, and this thing, this part is known as epicotyle. It is known as what? It is known as epicotyle. So student, I hope these diagrams are clear. Furthermore, if you have any questions or queries, you can definitely drop in. Otherwise, I would request you to go to page number 48 of ICSC Concise Biology Part 1 and follow the bean seed. We will be starting from there. So there are a number of different kind of beans such as broad bean, lima bean, French bean, etc. But in general, the structure of the seeds is the same. Most of the kidney shaped with a concave and a convex side. Okay, as we have already seen here. Now moving forward, we will get seed coat. Seed coat consists of testa, the outermost hard brownish covering. Okay, these are outermost hard browning covering, and it is known as testa. It protects the delicate inner part of the seed from the injury and from the attack of the bacteria, fungi and insect. Tegmen is the thin layer lying next to the testa and is also protective in nature. So see, the outer part is known as testa. See, this outer part is known as testa, and the inner side of that particular testa is known as tegmen. It also provides support. Okay, now the next is helium. What is helium? Helium is a distinct whitish oval scare in the concave side of the seed. It represents the spot where the ovule, now the seed, was attached to the ovary wall through placenta. So what happened? Helium is that distinct. This one is helium. This one is the helium. Okay, this one is the helium part. So what is helium? Helium is a distinct whitish oval scar on the concave side of the seed. Okay, there is a white scar on the concave side of the seed, and it represents the spot where the ovule was attached to the ovary through placenta. So it was the part from where ovule was attached to the ovary. It was attached to the ovary through placenta. Okay, and now it is developed in the form of helium. A tiny spore called micropyle is situated close to the helium. See, now this one, when we are opening up like this, we are getting this view, and in this view we find micropyle to be a small pore known as micropyle. Okay, it marks the opening through which the pollen tube had entered the ovule. Okay, now it is the mark through which pollen tube had entered the ovule, so it is now known as micropyle. Okay students, so micropyle serves two functions. What are they? When soaked in water, the seed absorb water mainly through this micropyle and make it available in the embryo for germination. And what is the second function? The second function is it provides for the diffusion of respiratory gases for the growth of embryo. Below the seed coat are two thick cotyledons which contains food for the embryo and protect it, and below that we have cotyledon which protects and give food and nourishment to the particular seed. On carefully separating the two cotyledons, the tiny embryo can easily be seen attached to one of the cotyledons. The embryo consists of two parts, the radicle which later turns to the root. So what happens? Radicle forms the root. It forms the root later on. Okay, and the plumule which later forms the shoot. And what happened? The plumule, it later forms the shoot. Okay, I am writing it down for your reference. This becomes the shoot. Okay, so I hope this is clear up till this seed, what is being seed and how the things are done. Next, the region of the axis between the point of attachment of cotyledon and the plumule is called epicotyle. The region of the axis below the cotyledons is called the hypocotyl. This is the radicle, this is the hypocotyl. Okay, so later on, this is all about the bean seed. We will go quickly to the maize seed. The diagram for the maize seeds are given in page number 49, figure 6.2. Okay, so please follow that diagram while we are continuing with the maize seed. The maize grain. The maize grain is actually a single seeded fruit in which the fruit wall and the seed coat are fused together to form a protective layer. Therefore, we call such a fruit as grain. So, what we have already learned about the definition of grain? We have already learned there the fruit wall and the seed coats are fused together, right? So, this maize is, we can classify it as a grain because the same thing happened here. See, the maize grain actually a single seeded fruit in which the fruit wall and the seed coat are fused together. Okay, so fruit wall and the seed coat, they are fused together in a maize grain. On one side of grain occurs a small light colored oval area which marks the location of the embryo inside. The remaining major part of the grain contains a large endosperm in which is rich in starch. The endosperm and the embryonic parts are separated from each other by a thin epithelial layer. The outermost layer of the endosperm is rich in protein and also called the aleurone layer. Okay. it is also known as aleurone layer, and it is rich in protein. The embryo consists of a single cotyledon here called scutellum, a radicle and a plumule. The radicle is towards the pointed end and it is enclosed in a protective sheath, the chlorosia. Okay, now you can follow the diagram, students, given in the next page only, and there you can get the exact details of what I am telling over here. So what you all will be doing is you will be listening to me what I am saying and seeing the diagram either in the board or in the book itself given. Okay. The plumule towards is the upper broader side of the embryonic region and is enclosed in a protective sheath, the cloeptile. Okay. So the male grain is a monocotyledonous and endospermic. Some other example of this type grains are rice, wheat and oat. These all are just like the maize grain. Okay. So we have a difference between maize and bean. I'll quickly go through this. It is available in page number 49. Okay. Now what is germination? The seed contains a dormant embryo. Now what is germination? Can anyone tell me what is germination? See, germination is nothing but it is the growth of the plant from seed. Okay, so we can call it that when a seed germinates, it forms a new plant. Am I clear? Germination. The seed contains a dormant embryo. In dry seed, the embryo is inactive. It is said to be in a state of dormancy or a period of rest. Outwardly, it appears without life, but in fact, all the chemical activities are going on it. Okay, so germination is the process of formation of a seedling developed from the embryo. So what is germination? Germination is the process of forming seedling from the embryo. This process is known as germination. So what are the conditions for germinations? Can anyone write that what are the conditions of germinations? Okay, so the condition of germinations are very simple. That is water, suitable temperature and oxygen. These three things are very much important. So I am rubbing this one. For any of the need, you can just rewind the video and get these diagrams. So what are the conditions? To briefly state, conditions are three. Number one is water, which is very important. Number two is suitable temperature. And number three is oxygen. Okay, so as we all know that water is obviously important and necessary, because without water it would not be possible for a seed to germinate, because as we already know that embryo contained in the seed, it is in a dry state and it needs water, because with the help of water it grows. Okay, the next point is suitable temperature. Suitable temperature is one of the very vital points, because it is the temperature which regulates the activeness of the particular embryo. Okay, so suitable temperature is needed for which or through which a seed can germinate. And third point is oxygen. As we all know, let's see student, I hope it is clear to you that plants do take in oxygen and take out carbon dioxide. Sounds odd, right? Yes. So what happened, we have already learned in our previous or from our parents or from the friends that plants give out oxygen and take in carbon dioxide, but it is only during the process of photosynthesis and not during the process of respiration. You need to be very clear on that, because plant take in oxygen and take out carbon dioxide during its own respiration, because it is a living being and in the cell these things are needed. But during photosynthesis, that is during the daytime, what happens is the chloroplast that is available inside the plant, they do take in carbon dioxide in a huge amount and it gives out oxygen in a very high amount, and thus we can say that during daytime plants gives out oxygen, but actually during germination or during its own respiration is not that the plant that gives out carbon dioxide, gives out oxygen. Okay, so you need to be very clear on that. Anyway, so oxygen is very much important. And one more thing, for suitable temperature we call it as a optimum temperature. Okay, optimum temperature. Now what is this optimum temperature? Optimum temperature is that it is neither too high nor too low. Okay, and we generally take 35 degrees centigrade to be the optimum temperature. Okay, 35 degrees to be the optimum temperature, but it varies from 25 degrees centigrade to 35 degrees centigrade. Now it depends on the weather, it depends on the condition, it depends on the type of seed that we are going for germination, but yet we can take 25 to 35 degrees to be the optimum temperature. So moving on, there are some experiments on germination. These experiments are very easy, you can go through it once. If you have any problem, I'll definitely go through these experiments. Also, you can do this experiment in your house out of fun. You can get a lot of a lot of experience from how the things are happening. And you know, practical knowledge is very much important. Without having the practical knowledge, it is like I'm going on studying, but I don't even know how to apply that particular study in our own state or in our own life. Now what is the point of studying? Say 90% of the students, you all have a question from your side. In fact, I do also have that same question, that so I am studying, ma'am, I am studying all these things, but what is the need of that particular thing in our practical life? We do not have that. So when I do a job, I go there, I work for a company, I get paid really well, I go to a market, I buy fish, but I don't even need all these seeds and everything in my life. But my dear student, you do need, because knowledge is the manifestation which is needed in you. Okay, without knowing how this seed is developing, how plants are coming on, see, there is a basic difference between an educated person and a non-educated person. A non-educated person does not even know that how these things are happening, but they know the things are happening. Now, as you have the opportunity, as you have got the thing, and you have opportunity, you have phone in your hand, you are opening YouTube channel, you are studying for a ICSE school, in a reputed ICSE school, you are doing everything. So why don't increase your knowledge? No, increasing one's knowledge doesn't mean that it do any harm, right? Rather, we can teach our next generation. Anyway, so there are these things. Now experiments, you can go through these experiments. For any of the problems, I will make you understand. Okay.